Hello, my name is James Cluxton. I'm a postdoctoral research fellow here at the Royal Botanic Garden, Sydney. And what I'm going to do is I'm going to give you an introduction to the cycads. Some of you will already have these plants growing in your areas, or you'll have some at home maybe, and you find these plants fascinating, or you're not that interested, but you want to learn a little bit more. But I think everybody hopefully will come out with something and watch this video and get an idea and more insight by the cycads, and hopefully bring up some talk and hopefully get some questions and answers, because these are incredible plants. And I've worked with these plants since 2008, and they're probably a personal interest. And I just find their morphology, and the more I work with these plants, it almost feels like the least I know. And I've loved these plants, and I think they are something to behold, and probably and are one of the most important plant groups that we can have in Australian botanic gardens. So let's start with a typical Australian species. This is Cycas calcicola. This species occurs in the Northern Territory near Katherine, as well as the Litchfield National Park. This is a quintessential Australian cycad, and it's growing in pretty much pure limestone. It's a really incredible plant. So let's talk about distribution, where we are at the moment. So cycads have pretty much a distribution that's below the Tropic of Cancer. The only difference is you see here, this is Cycas revoluta, which occurs in the Ryukyu Islands of Japan and coastal China. So in Australia, we have both families represented, Zamiaceae and Cycadaceae, and 4 of the 10 genera of cycads. As a note, within the Royal Botanic Garden, Sydney, and our collections as a whole, we have all 10 genera represented of the cycads, which is excellent for education and excellent for everybody to learn. So I really think that if you're interested in these plants, go and find out each one of these genera and have a look at them. They're awesome plants. Some of them are in the nursery, but we are finding a way to get these in the collections on display, so they have public interest there. So cycads, to me, really kind of home about something. They look a bit like a palm, but they have these big kind of pine cones on them. You've got this really typical stem. Not all species have this. Some of them have an underground stem, and they have a typical crown. You can see these like structures. You see these are caterpillars. The plants are either male or female. A lot of people will say, oh, my plants are male, or my plants are female. Well, they're not. No, a plant is neither male or female. It simply bears what I would say is mega or microsporangiate strobili. Mega being large, being female, micro being small, being male, and this is refers to gametes. That's it, and that's what's quite unique about them in that way. The dioecious, that's another way to kind of put cycads as well. So they produce caterpillars, and one of the things that they have is, for cycads, they don't always have a cone, which we'll go into a little bit more later, because some cycads you see is general, the females have a looser megasporphils, and you can kind of see them on this cycas on Botanic just here. And you have these distinctive mucilage canals. So you ever see a cycad that's, you cut a leaf off it, you'll see this resin protrude over it and harden. But if it gets really wet, it kind of oozes this like jelly-like substance. It's really poisonous, so don't touch it, or at least wash your hands afterwards. This is cycas, and cycas is a really cool genus. It is here. This is where we have the caterpillars. You can see this is on typical Cycas tatungensis or Cycas panzerensis. Just here we have this fluffy tomentum on the top of the stem. And you can see here, this is the leaflet. This is one of the key identifiers to Cycas. And this is these mid-veins on the leaflets. That's how you can identify Cycas. Cycas is also multipinnate. So it has tripinnate leaves or bipinnate leaves, you see. So leaves that are twice or 3 times divided, just like ferns. Now there's one more cycad that does this from Australia, and that is Berinia. However, there's no cycas in Australia that has this leaf. And if they do that, it's a little bit more unique. There are species of Macrozamia, actually, I should add, why I remember, that have quite unique leaf structures in what they call the Parazamia complex, these small growing Macrozamias. And cycas the new leaflets that emerge are called like watch springs, and they kind of unfoil, just like ferns, and it's really quite cool and quite unique. So when we look at the actual cone structure, this is the typical male cone that you find on cycads. And this is the female cone structure. Now you see I'm saying cone structure and not a cone, because there's something different here. A cone itself is connected to what you call a central axis, and you have the scales connected along this axis. Now, on the megasporophylls here, this cone structure on cycads are more loosely arranged. And this characteristic that you find on cycads is what I would say is this links this some earliest lineages of seed plants and plants that are physically producing seed. And this is the closest link we have to something like the extinct Benititales when you compare this to, say, pterophytes and the ferns. And here we can see this is the typical sporophyll structure. This is actually, I think, from Deberensis. And again, here, this is more megasporophylls from cycads, Meconicae in this case. And then we go to Zamiaceae. And this is where we talk a little bit more about morphological characters to separate these things. Just here, this is an encephalatus. In this case, I think this is Encephalatus princeps. And this is typical encephalatus leaf characteristics. And one of the things you'll find is you've got Zamia and Ceratozamia. Now, leaflet-wise, they look very similar, but there's one key difference. Ceratozamia have entire margins on the leaflet, whereas Zamia have dentate. Even if you look at Zamia pumila or Zamia integrifolia, you look very close, you'll feel slight dentation, not spiky, but just slight dentation like bumps. And Ceratozamia don't have that, and that's the best way to identify them just based on leaves. Diune leaves are quite straight and they're quite flat, and generally the leaflets are connected on top of the rachis. Lepidozamia have like a curl that goes in. I didn't have any images of the Lepidozamia. I would have put them in here, but I didn't have really free access to the garden to just go and photograph a Lepidozamia leaflet morphology. And then we have macrozamia. And one of the key identifiers of macrozamia is, look where the leaflet connects. So this is not the best image, but you can see these are the leaflets where it connects to this yellow bit just here. So macrozamia have this swollen callus where the leaflet connects to the rachis. And this is either yellow to reddy orange color. And if you remove that leaflet, more often than not, it tears away that callus at the end of the leaflet there as well. I mentioned Barinia have these multipinnate leaves, and then we have microcycas. And microcycas is really interesting. It's almost got a truncate leaf. The leaflets kind of reflects downwards, which makes it quite cool in that way. And then this is actually all of Zamiaceae. I've got cones for all of them. Just here we can see these distinctive cones from Encephalatus, Barinia, Diune, macrozamia in the spine at the end of the leaf here, Stangeria, microcycas, and this is Zamia, these really distinctive hexagonal shapes you get in Zamia, Lepidozamia. Now, we have two Lepidozamia species in Australia, Perovskiana and Hopii, and they are quite distinctive leaves. Generally, Hopii is much bigger. But if you look at the cones, when you look at Perovskiana cones get much bigger than Hopii, but the terminal bullae is abaxially downturned. So this terminal facet to the cone scale or megasporophyll, in this case, is downturned on Perovskiana and upturned on Hopii. And then Ceratozamia, you can't see them very well, but you see they're slightly spiky looking. If you look at Ceratozamia sporophyll, what you'll see is on males and females are these two horns, and what I would call bicornate spines that stick out from the cones, and that makes them quite unique. Cycads have a great appeal to the landscape and in horticulture, and this is what has really led to them being severely over-collected from the wild and things. They really resemble times gone by. They're very majestic. They're really, when you've got big planting of cycads, it really draws you to it. And they're just incredible plants. And they have some really unique relationships. So cycads have a unique push-pull pollination mechanism where the cones in a population can heat up in synchrony and then produce toxic chemicals to push beetles away. And why would they want to do that? Well, generally the male cones have all the nutrients and things and they're full of starch. The female cones, the female plant is putting all its energy into producing seeds. So it doesn't really put a lot of energy into the starch structure of the cone itself. So the beetles basically swarm around that, and then the male's cones can heat up to 12 degrees above the ambient temperature and kind of push the beetles away. And then the pull is where the females start to not heat up, but produce other chemicals. And it's all perfectly in sync. It's quite incredible. They have really unique pollinators, mostly beetles. So this whole thing about wind pollination is something to be considered. And it probably accounts for about 2 to 3% of pollination occurrences, which is not very high. If you look at an encephalatus female cone, think about it and look at the scales. When the scales start to separate and you can see movements like gaps, that's all they're going to open. Not going to open any more than that. When you think about that, wind isn't going to get there. And the pollen grains of cycads are not really small. They don't really carry on the wind. You can smell them, like cycads. When you're in the garden, you're walking around the encephalatus collection, the South African bed, there's some cycads planted around there. And when the males are in cone, they're really strong smelling and they really bring. So we know that, and one thing that cycads have is a relationship with cyanobacteria. They have these roots that protrude to the surface, quite strange looking roots that look like coal, called colloid roots. And if you pinch them, and if they've got this little blue-green layer in them, it means that's cyanobacteria. And that cyanobacteria has a relationship with the plant, and it allows the plant to actively fix nitrogen in nitrogen-poor soils through that cyanobacteria. And in exchange, the plant gives the cyanobacteria carbon and sugars. So cycads have huge economical importance, but population are heavily predated on. Predated, the wrong word. Heavily over collected and things for many purposes. And horticulture is one big significant factor, which has led to more than 62% of cycads being endangered, according to the IUCN Red List. And actually, it's the highest group, highest ranking of any organism on the planet, which is really significant. Cycads are also major horticultural crops. And they've been, especially from South Africa, like just here, this is Encephalatus limani. And this was a wild population, that has now, that basically got found, that was taken from the wild, and then it got somebody along the lines figured out this was a plant population that had taken, and Kirstenbosch in Cape Town were able to secure that population, and it will now go into their collection. These plants can't really go back into the wild because the soil conditions, they take so long to reestablish, but there's more to it than that as well. And cycads, of course, are used for medicinal purposes. There's no benefit to that. And of course, food, you've heard of sago. There is not really, there is sago and then there's sago. So sago from Cycas revoluta is different to sago from another type of, a type of palm. If there's a really good paper out there called The Island of the Colorblind, if you have a look at that paper, it's a book. And it's about people in the Ryukyu Islands who eat the sap of Cycas revoluta and get effects of colorblind and early Alzheimer's. It's quite significant. And cycads have, the reason is because cycads have a couple of toxins, 2 toxins which are endemic to cycads, which is cycazine and macrozamin. And the other toxin, BMAA, is the one that affects us when we eat the seed. There's a really good story of James Cook. They basically came to Australia, to, I think it was Northern Territory, and the indigenous people gave them some seeds to eat because they would eat them as a famine food. And it made the crew violently ill. They even said in the sentence is they was putting vile from both ends, so it doesn't sound good. And a load of the animals died on board the ship after on the way back. So, you know, don't eat cycads. So let's talk a little bit about the research that I'm doing at the moment. I'm just going to give you a bit of an idea. So I'm doing some previous work with Australian cycads, but one of the things I'm working with is the University of Johannesburg on encephalatus. This is one of the most threatening groups, and for poaching especially, and also it's a real collectors group. But there's no recent taxonomy treatments, there's no monograph, there's no recent revision. So the taxonomic knowledge on these plants is poor, and we need to understand it. But sometimes tackling um, the taxonomy from ground zero is very difficult and very intractable. And my opinion is that it's much better to understand and start from a genetic basis and then inform taxonomy based on genetics. This is exactly what this tree is doing here. This is a phylogenetic tree containing all published species to date of Encephalatus. And it's pretty good. It shows some good relationships, but there's a real good indications a lot of hybridization potentially happening within Encephalatus and the genus itself. And that's something that needs to be considered. And one of the things that botanic gardens can do is it's important that botanic gardens safeguard natural populations. And we know when botanic gardens can hold, say, over 200 individuals for the species, they can retain almost 80% of the genetic diversity for that species with over 200 individuals. It's a lot of plants for 200 individuals of a species, but it means 80% of the genetic diversity of the wild populations is a lot, and it's significant. And that's one thing that botanic gardens need to work towards, is to be able to, if they need to repopulate wild populations, or they can use these plants as educational values to teach, and that's something that's important. So for my own research on cycads calcicola, I showed you an image of it earlier. I did a conservation genomic study on this. So it's really looking at genetic diversity of the population. We found that genetic diversity was very low, very high levels of inbreeding, really high levels of decline in the populations. And what we found is ex situ populations did not represent the genetic diversity of what's found in the wild. So we can see this is a structure plot. So each individual block in here represents a species, an individual. The block is a population. One color is one genotype, another color is another genotype. So in cultivation, this is what we have: individuals from the Catherine region, and then we have individuals from Daly River and Spirit Hills, two regions that are not represented here. But look at this plot just here. From the Litchfield National Park, we already knew that it wasn't present in cultivation, but we now know that not is it not present in cultivation, but it's also genetically dissimilar from the others. So there's a huge portion of the genetic diversity missing. As a note, we do not have this species in our collections at present, but that will change. So what are the threats posed on the natural populations? And this is the one thing, as a botanic garden, we need to be thinking about conservation and science. And the threats posed on these natural populations are increasing ever more. And that's something we absolutely must and need to consider, because if we cannot always conserve natural populations. We have to have individuals represented in cultivation. And we can't, for groups such as Encephalatus, Zamia, Ceratozamia, we can have representatives in our collections, but we can't focus on holding genetic diversity, for example, for Encephalatus natalensis, because we can't go out and collect more plants from South Africa, especially. There are botanic gardens in South Africa that are already doing this. So we have to focus on what's important to us. But it still means that we still have to have these species within our gardens. We have 3 sites. We have Sydney, we have Mount Annan, we have Mount Alma, and each one has an indifferent microclimate. And this will allow us to grow different plants between these gardens. Of course, Mount Annan is Australian only, but Mount Alma and Sydney, we can have more diversity for, say, Encephalatus, Ceratozamia and Diune and things, and increase these. And we know that these collections have an incredible scientific value, and we have to maintain that scientific value, and also increase it by having plants of known wild origin. And the more plants we have of known wild origin, the better these collections really become for both scientific and horticultural research. And we really have to be at the frontier of horticultural research. And that's where we come into advertising the expression of interest for a botanical cycad horticulturist. And this is something that is very important because it means we have one person that can learn about these plants, attend conferences, carry out horticultural research on these plants, because we have to be a center for horticultural excellence. We have to be the best at growing these plants in Australia and be the experts at these plants on a horticultural level and a scientific level. Because if we want to be a world leading botanic garden in cycad conservation, cycad horticulture, this is the direction where we need to go. And we can do this, but we need to work together. We need to work on joint collection trips, go out in the field together, think about collecting new genetic diversity, target new species for cultivation, try all these new species, try new growing conditions, try new environments for these plants. And that's what we can do as a team and work together. Thank you very much. Hopefully you've learned something from this short presentation. And if you've got any questions, feel free to drop me an email or go to my website and have a look. You should also, you can also contact me on Twitter. You should follow me on Twitter because I'm amazing as well. But no, in all reality, if you've got any questions about this, I'm happy to answer it. And I really think this is an exciting opportunity. And I would love to meet anyone that's interested and to talk about cycads, because I love these plants and these plants are amazing. And these plants are a key part of our living collections moving forward.